Frederick Sanger. Frederick Sanger was born in England on August 13, 1918. He was named after his father, who worked as a medical practitioner. His mother, Cicely Sanger, was the daughter of a wealthy cotton manufacturer. Initially, Frederick Sanger was to follow his father's footsteps and become a medical doctor. However, after much thought, he realized his interest was in research and decided to become a scientist. In 1936, Frederick Sanger enrolled in St. John's College. Here he focused on chemistry and physics, however was later attracted to biochemistry. In 1939, he received his bachelor's degree but stayed another year to take an advanced course in biochemistry. He described himself as an above-average but not outstanding student. During his college years, Frederick Sanger met Joan Howe. They were married by 1940 and would eventually conceive 3 children together. After his marriage, Frederick was assigned as an orderly to a hospital near Bristol. Soon afterwards, he decided to go to Cambridge and enter the doctoral program in biochemistry. He studied lysine metabolism and analyzed nitrogen from potatoes. In 1943, Frederick Sanger would finally receive his doctorate. Afterwards, Frederick joined the laboratory of Albert Gimnall at Cambridge. Albert's group had previously determined which amino acids made up insulin, and Sanger's work on the structure was an extension of Albert's research. Frederick's first work was to identify the amino acid sequences of proteins by using a chemical that forms compounds with the acids and marks each with a different color. This is otherwise known as chromatography. From this method, he was able to identify 2 terminal amino acid groups by chemically breaking the sulfur bridges. He then found that insulin is composed of phenylalanine and glycine, suggesting that insulin is made up of 2 types of chains. From this finding, Sanger used the method to study the amino terminal groups of several other proteins. He used another method, electrophoresis, in which chain fragments move through a fluid at different speeds in response to an electrical current. He determined the amino acid sequence for each fragment and noted any common groupings. From this, he was able to compile the entire sequence of 21 amino acids for the A chain and 30 for the B chain. Frederick Sanger next worked on nucleic acids with American chemist Walter Gilbert. Whereas Walter's method preferred short segments, Frederick's method preferred long chains. Frederick had deduced the structure of insulin and was the first person to sequence a protein. For this accomplishment, Frederick Sanger was awarded the 1958 Nobel Prize for Chemistry. In 1962, the Medical Research Council opened its new laboratory of molecular biology in Cambridge. It was in this laboratory that Frederick was able to pursue studies on ribonucleic acid. The goal of this lab was to sequence a messenger RNA and determine its genetic code. By the time the sequence methods were worked out, the code had been broken by other researchers. However, Frederick's group was able to confirm the genetic code previously discovered. By the early 1970s, Frederick Sanger was interested in deoxyribonucleic acid, or DNA. He began using the enzyme DNA polymerase to make new strands of DNA from single-strand templates, introducing radioactive nucleotides into the new DNA. This would eventually lead to the first human gene to be sequenced. For this contribution, he received another Nobel Prize for Chemistry in 1980. Frederick Sanger decided to retire in 1983 to his home, which is located close to Cambridge. He has been honored with the awards of the Corday Morgan Medal and the Prize of the Chemical Society. He has been a member of the Royal Society, the American Academy of Arts and Sciences, and the American Society of Biological Chemists. In addition, Britain's Sanger Center, or a research organization dedicated to mapping and sequencing the human genome, was named in his honor. Needless to say, Frederick Sanger was a well-accomplished man and has made many contributions to further science research.